students, in previous class in NS subject I explained 4 factors which are essential to green plants, and also I explained why photosynthesis is considered as basic life process, and also I explained about autotrophic nutrition, and also I explained why green plants known as universal food providers. Already we learned about 4 factors which are essential to green plants to perform photosynthesis, that is external factors and internal factors. External factors are CO2 and sunlight. Internal factors are chlorophyll and water. If you will observe, this is the cartoon of photosynthesis. This is the cartoon of photosynthesis. If you will observe in this picture, we can find out a important external factor, that is sunlight. First one is, and important external factor, second one is carbon dioxide, and we can find out water, it is the internal factor, and chlorophyll, this is also a internal factor. Here 2 external factors and 2 internal factors, and a gas which released by green plants known as oxygen. So if you will observe in this picture, we can find out 4 factors. One is sunlight, carbon dioxide, water, and chlorophyll, and we can find out a gas which released by green plants known as oxygen. So this is the total picture which we can find out the factor which is essential to green plants. Now I will explain about another important experiment in 10th exam point of view, that is how to test presence of starch in green leaves. If you will observe in 10th class NS subject first lesson nutrition, 4 experiments will be for you. These 4 are very important in the exam point of view. In that, first experiment is how to test presence of starch in green leaves. This is the first important experiment in the exam point of view. We know that starch formation is the indication of photosynthesis. If starch is formed, we can say green plants perform photosynthesis. In this topic, I will explain about how to test presence of starch in green leaves or green plants. We know that starch means nothing but sugar or carbohydrates. If starch is formed, we can say that plants performed photosynthesis. If you want to write experiment, all we know that we have to follow some instructions. First we have to write aim. What is the aim of this experiment? This to test presence of starch in green leaves. That is the aim of this experiment, to test presence of starch in green leaves. Next heading is apparatus or materials required. If you will observe, to perform this experiment, we require some materials. What that materials means here? First one is test tube. Second one is soft and thin leaf. First one is test tube. Second one is soft and thin leaf. And third one is methylated spirit. Methylated spirit. Instead of methylated spirit, you have to take alcohol also. And fourth one is water. Fifth one is beaker. Sixth one is aluminium gas. Seventh one is Bunsen burner. Eighth one is tripod stand. What do you mean by tripod stand? The stand which have 3 legs, known as tripod stand. And last one is petri dish. And some chemical also required to test presence of starch, that is called iodine. Tincture iodine. These are the apparatus which required to perform this experiment. Again I repeat apparatus. One is test tube. Next one is methylated spirit or alcohol. Next one is leaf, a soft and thin leaf. Water, beaker, aluminium gauge, Bunsen burner, tripod stand, petri dish, iodine solution. These are the apparatus which required to perform this experiment. Next one is procedure. What is the procedure? Very simple procedure. Take a soft and thin leaf. Take a soft and thin leaf in a test tube which contain methylated spirit. Take a soft and thin leaf in a test tube which contain methylated spirit and boil the test tube under water bath. What is meant by water bath? We have to keep test tube which contain methylated spirit in a beaker which contain water. What is the reason for this means? If you will boil the methylated spirit containing test tube on flame directly, it may catch fire and test tube may burst. That's why we have to boil the test tube which contain methylated spirit in water bath. Water bath means we have to keep the test tube which contain methylated spirit in a beaker which contain water. Very simple procedure. Take a soft and thin leaf in a test tube which contain methylated spirit. Now boil the test tube under water bath. So procedure over. Next side thing is observation. What is the observation? After boiling, take out the leaf from the test tube with the help of forceps and keep it in a petri dish. Now add few drops of iodine solution. Add few drops of tincture iodine solution. So after that we can find out the leaf turns into deep blue or black. The leaf turns into deep blue or black. After that we can conclude that last reading is result. Based on this activity, I concluded that starch is present in green leaves. When you will add, after adding of iodine solution, if the leaf turns into deep blue or black, we can say starch present in the leaf. Last side heading is precaution. What is the precaution should be taken while doing this experiment? What the precaution means? You have to, already I explained, you have to boil the test tube under water bath only. You have to boil the test tube under water bath only. Why? Because methylated spirit is highly inflammable. If you will boil the test tube which contain methylated spirit directly on flame, it may catch fire and the test tube may burst. That's why you have to boil the test tube which contain methylated spirit under water bath only. This is the precaution. If you will observe, these are the side headings are very essential to write this experiment. You have to draw the diagram also. If you won't draw the diagram, you will not get, may lose 1 mark. You have to draw the diagram compulsory in the exam point of view. Okay, first one is aim, apparatus. After that you have to draw the diagram. After that you have to write the procedure, observation, result, precaution. Sometimes in the exam point of view they will ask like this. They only will give diagram for you. No need to draw the diagram on that time. So they only will give diagram for you and they will ask some questions below the diagram 1, 2, 3, 4 like this. First they will ask like this. What is the aim of this experiment? This is very easy. No need to draw the diagram on that time. What is the aim of this experiment? To test presence of starch in green leaves. Second question they will ask like this. What are the apparatus used to perform this experiment? Test tube, methylated spirit, leaf, water, beaker, aluminium gas, Bunsen burner, tripod stand, iodine solution, petri dish. Third question they will ask like this. Why we have to boil the leaf in methylated spirit only? Why we have to boil the leaf in methylated spirit only? This is helpful to remove chlorophyll. To remove chlorophyll in the leaves. This is very essential to test starch very easily. Fourth they will ask like this. What is the precaution should be taken while doing this experiment? You have to boil the test tube under water bath only. 4 lines answers will get 4 marks. So first aim, second one is procedure, third one is why we should boil the leaf in methylated spirit only, to remove chlorophyll in the leaf. And the last one they will ask what is the precaution should be followed while doing this experiment. What the precaution? You have to boil the test tube under water bath only. Why? Because methylated spirit is highly inflammable. Now Again I repeat the same experiment. Why? Because it is very important in the exam point. Aim to test presence of starch in green leaves. Apparatus: test tube, methylated spirit, leaf, water, beaker, aluminium gas, Bunsen burner, tripod stand, iodine solution, petri dish. Procedure: take a soft and thin leaf in a test tube which contain methylated spirit. Now boil the test tube under water bath. Observation: after take out the leaf from the test tube, keep it in a petri dish with the help of forceps. Add few drops of iodine solution. The leaf turns into deep blue or black. Result: based on this activity, I concluded that the starch is present in green leaves. Precaution: boil the test tube under water bath only. Now I will explain about factors which are essential to green plants to perform photosynthesis. First factor is water. Water is very important internal factor which is essential to green plants to perform photosynthesis. The importance of water to green plants is explained by a scientist known as von Helmont. He is a scientist who conducted some experiments on green plants and he revealed the importance of water to green plants. Von Helmont is the scientist who explained water increase the mass of the plants. Mass of the plant. Mass means here weight. The water increases the weight of the plant. He conducted one experiment to reveal the importance of water to green plant. For this he selected a plant and he observed the weight of the plant and he also observed the weight of the soil and he poured water to green plants. After that he observed, after some days he observed the weight of the plant and weight of the soil. After that he noticed that the weight of the plant increases and the weight of the soil decreases. Based on this, he concluded that, von Helmont concluded that the plant mass will be increased with the help of water. With the help of water. So, first factor is water. Second internal factor is chlorophyll. Chlorophyll is the one of the important internal factor which is essential to green plants to perform photosynthesis. Do you know chlorophyll is a green color pigment present in a special organelle called chloroplast. Already we learnt in 9th class in plant tissues we learnt about chloroplast. It is a type of organelle which present in green parts of the plant known as chloroplast. It is present in a tissue known as chlorenchyma. We learnt in 9th class. So, without chlorophyll, green plants cannot perform photosynthesis. So, to explain about the importance of chlorophyll, you have to take a leaf known as variegated leaf. A leaf known as variegated leaf. What is meant by variegated leaf? The leaf simply called as croton. Croton leaf, croton plant. If you will observe in variegated leaf, we can find out two regions. One is chlorophyllous areas and second one is non-chlorophyllous areas. What is meant by variegated leaf? The leaf which contain different shades. Instead of green color, we can find out different colors. That is known as variegated leaf, simply called as croton leaf. These plants are grown in your houses for decoration, simply called as ornamental plants. So if you will observe, we know that leaves will be green in color. But if you will observe some leaves, we can find out different colors instead of green leaves. Green color, that is known as variegated leaf, simply called as croton leaves. So you have to take a croton leaf from a variegated plant and this leaf must contain chlorophyllous areas and non-chlorophyllous areas. Chlorophyllous areas means which have green color. Non-chlorophyllous areas means which does not have green color. So you have to keep the leaf in a test tube which contains methylated spirit. Already I explained in the previous So you have to keep this leaf in a test tube which contains methylated spirit. After that you have to add some iodine, some drops of iodine solution to the leaf. What will happen if you will observe like this? If you will observe the leaf which have green color turns into deep blue color. Here this is deep blue color and the leaf which does not have green color turns into pale color. The leaf which does not have green color turns into pale color. So the chlorophyllous areas turns into deep blue color and the non-chlorophyllous areas turns into pale color. What is the reason? Here we cannot find out any chlorophyll here. That's why the part which does not have chlorophyll is simply called as non-chlorophyllous areas turns into pale color and the chlorophyllous areas turns into deep blue color. Based on this activity, we concluded that chlorophyll also very essential to green plants to perform photosynthesis. Without chlorophyll, green plants cannot perform photosynthesis. That's why the part which does not have chlorophyll turns into pale color due to the absence of starch. So based on this, we concluded chlorophyll is the internal factor which is essential to green plants to perform photosynthesis. In the next class, I will explain about importance of air to green plants to perform photosynthesis. Thank you.